Eyepiece or ocular lens, located on the top of the body tube, magnifies the specimen 10x, while the objective lenses, located on the nose piece, at the bottom of the body tube, and used in combination with the eyepiece, to provide a range of magnification. Furthermore, objective lenses 8x, or 10x, take, shortest lens, and 100x take, longest lens, in normal. Therefore, 8x, obtain, initial focus on a sample, to scan, a slide, for a point of interest. Meanwhile, 10x, examine any finding closely, like mitosis and meiosis divisions, whereas, 40x, examine any finding in a greater details. In addition, 100x, examine in more details the findings. So, for scanning and low power, use coarse and fine knob, and for high power, use only fine knob. Nose piece, located at the bottom of the body tube, so, it holds the objective lenses, and rotates to enable magnification. Arm and base, are used to carry and support the upper parts, and whole body of the microscope, by adding, one hand on the arm, and the other hand, on the base. Stage, is used to support the slide, which contains specimen, or, object, that are viewing with the microscope. Stage clip, helps to hold the slide, in place, to allow light to pass from the light source to the lenses. Light source, provides light necessary for viewing the specimen, by sending the light through the stage opening, to the diaphragm. Diaphragm, is a wheel or lever located below the stage opening, used to regulates the amount of light, that can enter the lenses. Coarse adjustment knob, raises and lower the stage or objective lenses, and used only when focusing the low-power objective lens. Fine adjustment knob, raises and lowers the stage or objective lenses, as small distance, for exact focusing, and used when focusing the medium-power and high-power objective lenses. This slide present, the image plane, passes from the objective lens to the eyepiece lens.